So hi everybody, my name is Amanda Bataycan. I'm a UTEP PhD student. I work under Dr. Long and Dr. Mole, and today I'm going to be showing you the application of using my devised integrated cumulative scoring function and its application on non-synonymous single nucleotide variants in patients with acute leukemia. So before I begin, I'd like to acknowledge the grant that actually funds my work and thank them for their continued support. So just to get a brief background on acute leukemia, there's two different types, lymphoblastic and myeloid. So acute lymphoblastic leukemia occurs in lymphocytes, whereas acute lymphoid leukemia occurs in myeloblasts, red blood cells, and platelets. Now, both are acute forms of leukemia, so that means that their mutations are arising in early cell stages, and it makes them actually an aggressive form of leukemia. Now, in order to conduct this research, I extracted VCF files from the GDC, and from there, I isolated the non-synonymous variants that are occurring only in protein coding regions. And additionally, since I'm trying to figure out the novel genes, I've had to compile a list of associated genes to both ALL and AML. So that's actually going to be used later on. And there is a table with all the different counts. I won't go through and read the numbers to you because they're big numbers, but just so that they're there. Now, just to get a kind of idea of like the levels of this cumulative scoring function, I have here an extracted image from Ensemble, and it actually shows the overlapped genes KRAS and ETFRF1. So the horizontal lines are all of the transcripts that occur between both of these genes. And I've placed actually these vertical purple and red lines that show hypothetical variants that we're going to look at. So if we're looking at the red line, it's the very first one. And what it is, I think this has a pointer. Maybe not. But anyways, so the red line is actually going to show the red variant. So this is a variant that has the potential to affect both genes. It's the KRAS and the ETFR, and it also intersects at least 10 transcripts. Now, for the purple line, it only intersects transcripts found in KRAS, but it has 12, so that's on that level. Now, to avoid my research looking like a children's book, there's a different way instead of just classifying these variants as red and purple. What we use are variant effect analyzers. So for my research, I've used 4 different ones. It's going to be Fathom, CAD, SIFT, and Polyfen. Now, what these do is they quantify what a variant is. So it can determine if it is deleterious or not. And what it inputs is the chromosomal position and the mutation and returns a certain score for each variant. So that's what's going to be used here. Now, this is the cumulative scoring function, which we've called QG. What this does is it's a summation for each gene that takes into consideration, or it sums up all of the alterations between the transcripts and mutation level for the variant. So there's this, and pretty much the interpretation behind it is, since it's a cumulative scoring function, the higher the score, the more likely it is going to be associated to its corresponding leukemia. Now, since we're dealing with 4 different effect analyzers, they all have different score ranges. They have different prescribed cutoffs. So in order to unify and be able to integrate these results, we have to actually do a normalization process. So this is the formula that we have here. And then the table is just showing the different scoring ranges as well as its cutoff. So some actually look at the numbers that are higher, and then SIFT is the only one that actually looks at numbers that are lower. So the lower the number of the score is, the more deleterious it is. Now, to look at the gene correlations among all 4 effect analyzers, what this does is it determines the relationship between all 4. So this is actually an output from an R package. It's a performance analyzer. And we're using the Spearman method, so in case you guys are curious about that. But really what we want to look at is if the correlation coefficient, which is in the top right-hand triangle, if it's closer to 1, it means that the scores are more identical to each other. But our scores are nowhere near 1. They're like somewhere in the middle, which doesn't necessarily mean it's a bad thing. What this shows is it's showing variation among the different effect analyzers. So that's actually useful because If all four scores are saying that a variant's deleterious, then it's more likely going to be deleterious. However, if we have one that's saying it's deleterious and three others that's saying it's benign, then we want to take that into consideration. So we're actually glad that we have this variability among the different effect analyzers. Now, after we've done the normalization, we can now integrate these scores together. So this is the integrated cumulative scoring function. So I've labeled this as the IQ gene instead. Now, what this does is it takes the average among the four different effect analyzer scores, and then it uses that instead of just your traditional scoring method. In this table here at the very bottom, this is just a sample of inputted information. So the first few columns, one, two, three, four, the first four columns are actually variables that are inputted into each effect analyzer. So that's where we get the scores from. We also have the gene column, the transcript column. So that's how we're able to identify which variant goes to which transcript and gene. And then the following columns are the different scores. So this is just sample output. And then the very last column itself is the integrated score. So it's just the averaged among the four. So that's just a sample. Now, these are some inputs that were used because we're all about numbers. So these are just numbers of the inputs. And from what you can see here, we have thousands of genes from each effect analyzer. Some are different numbers. This is because some effect analyzers don't return an output for certain things. But that's the benefit of using an integrated scoring method, is it encompasses, I guess, all of the effect analyzers. So that's why the integrated score has the highest number, because it's taking information from each one of them. So that's the benefits of using this one. And then in parentheses is actually the number of transcripts. So our formula goes based off of the transcript level to the variant mutation level. So that's why it's important information there. Now, as I mentioned before, we've compiled a list of known genes. So for both AML and ALL, there is a list of known genes from previously published work, from different databases. So all of that has been compiled into a single list. And what we want to look at is the overlap percentages. So from our IQ gene score, we want to see which ones are actually known and then investigate the ones that are unknown. So what this shows is it has it for the four different effect analyzers, but the purple column is our integrated score. So it's not the best of the best. However, it is competitive against the other four scores. We don't want it to be exactly perfect to where it's showing 100 across the board, because then we won't be able to see the other genes that could be associated to AML or ALL. So that's just what this is showing here. Now, the IQ gene score, like I mentioned before, it takes into consideration the mutational variant level. So that's what the tumor minus the normal sample is. So our VCF files, we have both normal and tumor samples. And so what would happen if we were to remove that mutational difference? So this is exactly what will happen. The line graph here actually shows a solid green and a dotted green line. The dotted green line is actually the one without mutational information. So it's just setting that value equal to 1. And as you can see, there is a difference between the two. It does lower our overlap frequency. So we do want to have that mutational data. It does show that it has like a benefit for it. This one here is just for ALL, but it's the same trend observed in AML as well. So the SNV frequency does give that necessary weight to the equation. So we have thousands of genes, as you see, but I won't list all of those, but I will show you the top 15. So these are the top 15 Q gene scores for both ALL and AML. Now, in red is the ones that are novel, or the ones that aren't found in any published literature. So those are the ones that we want to kind of take a deeper dive into. As you can see, there's the transcript level, or the number of transcripts per gene. There's the number of variants per gene, and then the score itself. There is a difference between ALL and AML. ALL has a much higher Q gene scores. That's what I believe to be because there's a higher sample size. So the previous table in the way back beginning, it did have over 600 files, whereas AML only had 182, I believe. So to take a deeper dive into those novel genes, the ones that are highlighted, we just kind of looked through the top three. I won't read everything off of this table, but I will let you know that those genes are actually associated to other diseases and cancers. So, and then also within this table, we have the rank on where they occur within the output itself. For future work, we plan on taking a deeper dive into the genes that are outputted by doing a protein-protein interaction network. There's similar softwares, I think dbstring and other ones that we plan on using. But also we are closely affiliated to the pharmaceutical department at UTEP. So I plan on pushing those novel genes over to them so that way they can use their own different wet lab studies. So that's what I plan on doing. And thank you guys. Hopefully that was fast enough and I didn't go over my time. All done? Oh, there's a poster. If you guys want to look at the ovarian cancer results, A19, that's going on at 12 today.
Right, so, yeah, Angela, please, yeah. So we might have time for one quick question if anybody has one. Maybe squeeze in one. We are really running through. Oh yeah, go ahead. If you could come up to the mic, please, quickly.
Is that fast enough?
That is perfect.
Okay.
Hi. How do you think how confident are you in your results if you would change your effect predictors, like add some, remove some, switch them out?
Still fairly confident. I mean, that's the beauty of having the integrated scoring formula is I get to be able to use all these different effect analyzers. So in this case, I think more is better because some effect analyzers look at only the autochromosomal ones, some look at other ones. So I think for Fathom XF, that one is one where it doesn't really look at the X or Y chromosomes, but I've had research where we should focus on like the Y chromosome for AML, but so that's why I like having to incorporate all four, but I think having more would actually be beneficial, but it's a lot of work, so.
Thank you.